Hello, and welcome to the American Association for Cancer Research's press conference previewing the AACR Virtual Annual Meeting 1. I'm Julia Gunther, Associate Director of Public Relations for the AACR. Thank you for joining us today. The goal of this press preview is to offer a glimpse into the innovative science to be presented at the AACR Virtual Annual Meeting 1, happening next Monday and Tuesday, April 27th and 28th. Our esteemed presenters will also discuss the impact the COVID-19 pandemic is having on patients with cancer and the field of cancer research, and offer a look ahead to the second virtual annual meeting to take place June 22nd through 24th. We will conclude with a question and answer session. You may submit your questions at any time during the webcast using the Q&A box at the bottom of your screen. I will read the questions during the Q&A portion, and Drs. Martis and Rebus will answer. Please include your media outlet and if your question is for one or both of our speakers. Before I formally introduce our speakers, I'd like to share a couple of other housekeeping items. First, the embargo for content in today's press preview lifts at 12:01 a.m. Eastern Time on Monday, April 27th. This coincides with the posting of abstracts for the AACR Virtual Annual Meeting 1. Second, everyone who registered for this press preview has also been registered as press for both virtual annual meetings. You should have received an email from the AACR with information about accessing the sessions on April 27th and 28th. If not, please contact our media relations team. And now it's my pleasure to introduce you to our two distinguished presenters for today. Elaine Martis is the president of the AACR. Dr. Martis is co-executive director and endowed chair of the Steve and Cindy Rasmussen Institute for Genomic Medicine at Nationwide Children's Hospital in Columbus, Ohio. Antony Rebus is the program chair of the AACR Annual Meeting 2020 and also the AACR president-elect. Dr. Rebus is professor of medicine, surgery, and molecular and medical pharmacology at UCLA Medical Center. Dr. Martis, Dr. Rebus, I'll turn this press conference over to you.
Good morning, everyone. Dr. Rebus and I want to thank you for joining us today and for all the important work you do to cover cancer research news. I hope that you are all safe and well wherever you are around the world. It's my great honor to welcome all of you to this preview of the first AACR virtual annual meeting. We are living in unprecedented times, and this is certainly not the press preview I had imagined for the AACR Annual Meeting 2020, which Dr. Rebus and I started planning with the help of our esteemed organizing committee and the AACR staff almost a year ago. But the rapid escalation of the COVID-19 pandemic in the United States in February and March of this year made it necessary for the AACR Board of Directors to terminate the annual meeting scheduled for April 25th through April 29th in San Diego. This was a difficult decision, but absolutely the right one, because it was imperative that we do our part to safeguard the health and safety of everyone who was planning to attend the meeting, and ultimately that of the patients and communities they serve. Initially, we had hoped to hold an in-person meeting later in 2020, but it soon became clear that this would not be possible. Instead, the AACR Board of Directors made the decision to convene a second virtual meeting from June 22nd to the 24th. This combination of virtual meetings will permit rapid sharing of new results that would have been presented during the in-person meeting, and will disseminate cutting-edge data that fosters collaborations, sparks innovation, and drives progress for cancer patients. Working together with my colleague, Dr. Antony Rebus, the program chair for the AACR and the AACR president-elect, with Dr. Margaret Foti, CEO of the AACR, and the AACR staff, we have adapted the program that was planned for the in-person meeting to create two virtual meetings. The first of these is taking place next week on April 27th and 28th, with more than 26,000 attendees registered from 103 countries. We're anticipating more than 30,000 attendees by next week. The second, as I mentioned, will be a 3-day virtual meeting in June from the 22nd through the 24th. Dr. Rebus will give a comprehensive overview of the program for the first virtual annual meeting in a few minutes. Its content largely focuses on late-breaking results from clinical trials. Yet, because these trials build upon discoveries from basic and translational research, sessions showcasing high-impact advances in these areas also will be included in the first virtual meeting. We are also working to ensure that the program for the second virtual meeting in June captures the exciting research being conducted across the breadth and depth of the cancer field, from cancer biology to population science, to prevention, early detection and interception, to treatment and survivorship research. Although these virtual meetings are taking place at a challenging time for everyone, I have found it important to reflect on how extremely fortunate I am to have served as the president of the AACR during this incredible time in the history of cancer research. The pace at which we are making pioneering technological advances and groundbreaking scientific discoveries across the spectrum from basic to clinical cancer research has never been more rapid or more broad-based with respect to interdisciplinary science. As we have delved deeper into the complex biology of cancer, we have gained a greater scientific understanding of how cancer arises and develops. Translation of this knowledge into clinical and public health advances is leading to unparalleled progress in cancer prevention, detection, diagnosis, treatment, and increasingly to cures. For example, in the United States, this remarkable progress is illustrated by the fact that cancer mortality has fallen by 29% since its peak in 1991. In effect, this decrease translates to 2.9 million fewer cancer-related deaths. Despite the remarkable progress, we know that the public health challenge posed by cancer is growing due to many factors, including the increasing incidence of obesity, challenges due to healthcare disparities in our minority populations, and comorbidities associated with cancer that occurs at an early age and lead to poor quality of life and secondary cancer susceptibility in survivors. It is expected that the number of new cancer cases diagnosed in the United States alone will rise from about 1.8 million this year to more than 2.3 million in the year 2040. It is clear, therefore, that there is an urgent need for continued transformative research. Here to tell you about how our programs will feature these new advances in innovative cancer research is our program chair for the AACR Annual Meeting 2020, Dr. Anthony Rebus. Tony is also the president-elect of the AACR, and I'm very much looking forward to continuing to work with him during his term as president to ensure that the momentum we have built this year is maintained. Tony is an internationally recognized physician scientist who has been at the forefront of cancer precision medicine and immunotherapy for many years. He serves as professor of medicine at the David Geffen School of Medicine at UCLA, as director of the UCLA Johnson Comprehensive Cancer Center Tumor Immunology Program, and as director of the Parker Institute for Cancer Immunotherapy Center at UCLA. Please welcome Dr. Anthony Rebus.
Thank you so much, Elaine, for your fabulous introduction. It's been wonderful working with you to develop the virtual annual meeting program. Hello, everyone. And again, we thank you, the members of the media, for joining us today. We truly appreciate all you do in your roles as journalists for cancer research. Let me begin with a brief overview of the program of the AACR Virtual Annual Meeting 1. This meeting has been organized in just 1 month to present the data that required immediate release and represents a first-of-its-kind large cancer virtual meeting. We decided to focus largely on clinical trials for the first virtual meeting because we believe it is crucial that these data are shared rapidly to ensure that they provide the maximum benefit to patients. The program will include clinical trials in the form of plenary sessions and posters, first disclosures of innovative small molecules and biologics that have been recently entered phase 1 clinical trials, and mini symposia that will feature basic and translational research presentation. The first drug disclosures will be featured in 3 new drugs on the horizon special symposia. There are 8 clinical plenary sessions that showcase the data from several exciting clinical trials. The sessions will feature expert discussants who will provide perspectives on the science behind the clinical trials being presented. The opening clinical plenary session will have 4 presentations of high-impact clinical trials, all of which were designed based on advances in preclinical research. 3 of the studies include a combination of targeted therapies and immunotherapies, one in breast cancer, the other one in prostate cancer, and another in melanoma. These combinations are based on preclinical rationale where targeted therapies induce a specific killing of cancer cells, and enlisting an immune response by giving an immune checkpoint inhibitor could make these responses durable. The I-SPY breast cancer study demonstrated that the triple therapy of a targeted agent, an anti-PD-L1 antibody, and chemotherapy was better than chemotherapy in shrinking cancer when given before surgery. The phase 3 randomized melanoma study called INSPIRE 150 demonstrated that the triple therapy of 2 targeted drugs and an anti-PD-L1 antibody was better than 2 targeted drugs for patients with BRAF mutated melanoma. This is a practice-changing study for this indication. The opening plenary will also include another melanoma study that tested the concept of intermittent therapy. The idea is that giving an anti-cancer targeted drug with an intermittent dosing may avoid the cancer getting used to it and becoming resistant. This concept had been developed in preclinical models and taken to the clinic within the National Cancer Institute-funded U.S. cooperative groups. Despite the solid scientific rationale, this randomized clinical trial demonstrated that continuous dosing was better than intermittent therapy with BRAF and MEK inhibitors. I would like to highlight 2 presentations in the liquid biopsy plenary session. They both provide information on studies involving thousands of patients using liquid biopsy assays for the early detection of cancer. These 2 presentations highlight that analyzing blood samples, investigators were able to detect cancers before they would have been otherwise diagnosed. Not only this, the blood assays also predicted the site of origin of the cancer. This is a rapidly evolving field, and this session is providing a glimpse to the immediate future of cancer diagnosis and management. The plenary session dedicated at CAR-T gene-modified cell therapies will include 2 presentations on next-generation products that are already being tested successfully in the clinic. One will use a universal or allogeneic T-cell product, which is manufactured using CRISPR-based technology and provided a high response rate in patients with leukemia. This approach would allow making the cell therapy more broadly applicable by shortening the manufacturing and making it cheaper. An equally high response rate in leukemia was achieved in another study using a bispecific CAR construct that is designated to avoid relapses by mutations in the target, which is a limitation of current approaches with single CAR-T constructs. Finally, I would like to highlight presentations in the lung cancer plenary session. One of these studies uses a liquid biopsy approach for the early detection of relapses of lung cancer after surgery. Investigators showed that they could detect relapses 6 months earlier than would be available by using conventional approaches. I want to also highlight a public health and disparity study on the geographical location of vape shops selling e-cigarettes. Investigators provide evidence that the vape shops are more frequently located in neighborhoods of low-income and higher minority populations. There are many other interesting and important presentations in the clinical trials plenary session. I hope you enjoyed the program. I will now turn it back over to Elaine to talk about the mini symposia that feature basic and translational research.
Thank you, Tony. We wanted to acknowledge the critical importance of foundational, basic, and translational research that leads to the clinical advances you'll be hearing about by selecting mini symposia on these relevant topics that feature new advances to balance the clinical trial sessions. The featured basic research is key to making clinical advances and includes new findings regarding cancer vulnerabilities and therapies, drugging undruggable targets, immunotherapy biomarker refinements, new cancer prevention translational efforts, and new genomics and microbiome-based studies of cancer. With the current onset of the COVID-19 virus across the world, I will now invite Tony to describe a session that he originated for this meeting that is going to focus on COVID-19 and cancer and the path forward with this viral disease and its impact on cancer researchers at this time. Tony, could you give us a little information about the COVID-19 and cancer session, please?
On the second day of the meeting, we're going to open the sessions with a COVID and cancer plenary session. We have invited cancer investigators that have been at the epicenter of the pandemic and have captured scientific information on the effects of COVID-19 in patients with cancer. We will have reports from Wuhan, Northern Italy, France, and Spain, focusing on the effects of COVID-19 on cancer care, as well as information on COVID and cancer from the highly diverse population from New York City. Furthermore, we will have a presentation on the use of oncology drugs repurposed for the treatment of COVID-19. I am excited with the timeliness of sharing this information with the AACR community.
Thank you, Tony, for that important overview. We will finish up our press preview session now with a brief mention of the contents of the AACR Virtual Annual Meeting 2 coming up in June. This meeting will feature our originally planned opening plenary session that explores the range of impactful cancer research and clinical translation. There will be thousands of proffered papers in mini symposia and in e-poster platforms, as well as selected sessions from our originally planned meeting content. Although we were not able to feature every session that normally is offered during the annual meeting, we did strive to provide the normal breadth of areas of expertise that reflect the interests of our AACR membership. This meeting will also include Distinguished Public Service Award lectures from individuals who have made extraordinary contributions to the cancer field, as well as a special lecture from NCI Director Dr. Ned Sharpless. In addition, the meeting will feature a comprehensive educational program featuring about 70 educational sessions and methods workshops. This concludes our press preview. Thanks so much for joining us. We will now transition to the Q&A session.
Thank you both. As a reminder, if you have a question, please type it in the Q&A box at the bottom of your screen. Make sure to include your media outlet and if you're directing your question to Dr. Martis, Dr. Rebus, or both. Okay, looks like we have our first question from Lauren Pecorino. And she asks, I think this could be for both Dr. Martis and Dr. Rebus, will the exciting findings on extrachromosomal circular DNA be covered?
Yes, so this is a recent research from Stanford University. The head of that laboratory, Howard Chang, is one of the speakers in the opening plenary session for the virtual meeting 2. He had been scheduled to give this open planning session this weekend if it hadn't been for the COVID-19 and pandemic and having to terminate that meeting. So that opening plenary session has been moved to the June 2nd virtual meeting.
Thank you, Dr. Rebus. And as a reminder, if you are submitting a question, appreciate if you would include your media outlet in your question as well. Our next question is from Lynn Peterson with Trends in Medicine, and Lynn asks Dr. Rebus, Dr. Rebus, could you comment on the outlook for menin inhibitors?
And I'm sorry, I will not be able to answer this question. I don't know about menin inhibitors. Could you expand?
Len, if you could send us a bit more information, we will try to get your question answered shortly. We have a question as well from Kate Yandel at Cancer Today. She asks, and I'd like to present this to both Dr. Rebus and Dr. Martis. What needs to happen before multi-cancer early detection tests can be used routinely in the clinic? And what will be the challenges in getting these tests to that point?
So I can start with this. As you've no doubt noticed, and as Tony highlighted in his remarks, we do have sort of late-breaking results, both from a study conducted across multiple cancer centers with the commercial entity GRAIL, as well as the DETECT-A assay coming out of Johns Hopkins University and Bert Vogelstein's laboratory. So these are sort of first of their kind early cancer detection approaches. They each have their own similarities and differences. So I'm sure you'll be interested to hear more from the speakers on these particular approaches and the results that have been obtained so far. I think to address the question in terms of how soon we'll see these different modes of early detection entering the clinic really comes down to a number of questions that will be discussed by these presenters, including, importantly, the comparison of false negative and false positive results that are obtained, and sort of the downstream consequences of those and what that entails in terms of the healthcare modalities and costs associated that are invoked. So I think it's early days, but we were really excited to see both of these abstracts preferred into our meeting, and I think you'll be able to learn a lot more from hearing these presentations about next steps. Tony, did you have anything to add on that?
Yes. I think this is one of the areas of cancer research that's most rapidly becoming reality in the clinic. These two abstracts present data on tens of thousands of patients, one 10,000, the other one 15,000, that had a suspicion of a cancer. And with a blood test, they could not only confirm or not confirm that there was a cancer, but also know where it originated. And that was before many of these cancers would be diagnosable otherwise. This is something that we're seeing the future in real time. I think this session will be remembered as when we saw the big data that says this is there and these assays should be broadly applicable, provided that they have the appropriate regulatory agency approvals. But it's all there. This will not take that long that will change our practice and diagnose with a blood test.
All right, thank you both very much. I have a question from Vivian Marks with Nature Research. And Vivian asks both of you, how are clinician researchers integrating the evolution of cancer in terms of genomes, epigenomes, microbiomes, et cetera, when treating patients?
So great question. There are many answers to it. I think one of the areas that's certainly been enabled in this regard is the sort of the second half of the session that we were just commenting on. The first half, of course, is this question about early detection. The second half, as well as some of the other sessions, are covering aspects of what we're commonly referring to as liquid biopsy. So using the genomics of a tumor or specific mutations in particular to sort of follow that patient over time in a less invasive, rapid and less expensive way than conventional imaging. And this is really one of the areas that I think is emergent in the sort of clinical translational space, to be sure. The other component of that, of course, is looking at immune responses, again, monitoring from the blood. I think in particular, as well as some other bodily fluids, is sort of entering into the clinical reality in some specific types of cancer. I also wanted to point your attention to the session on evaluating the genome and epigenome for more information about what's happening in this space. In particular, this session evaluating cancer genomics from normal tissues through evolution to metastatic disease has a number of nice presentations that further address how, for example, in the abstract from Memorial Sloan Kettering, oncology is sort of being pursued, or sorry, clonal hematopoiesis is being pursued in terms of the transition from pre-malignant to malignant disease, and as well some interesting papers on sort of longitudinal sampling, so looking at cancers over time and space to really understand the impact of therapy, the impact of genetics on progression into cancers as well. So lots in that space in terms of the clinical translation of genomics and treatment.
Thank you, Dr. Ribas. Okay, we have some more information from Len Peterson with Trends in Medicine. Len, thank you for sending that clarification. She's asking specifically about one of the new drugs on the Horizon sessions and the Menin and Augment 101 trial update. I don't know if Dr. Martis or Dr. Ribas, you'd like to comment on that at this time.
And I'm sorry, Lynn, because I show my ignorance in this field, and the new Drugs in Horizon session is one where the review committee, neither Dr. Martis or myself were part of the review committee. There's a specific review committee at AACR that looked at those abstracts and proposed them. So I don't think either one of us are prepared, but I'm sure through AACR staff, we can put you in contact with the first author and answer all of your questions with a lot more knowledge than either one of us.
Thank you, Dr. Ribas. All right, we've got a question from Raj Gopinath at Gene Online, and he asks, what progress have we made in CAR T therapies for solid tumors, specifically with regard to targeting the MUC1 inhibitor? Dr. Ribas, would you like to answer that?
Yes, so there's in the from what I can recall in the oral session, there's not a MUC1 CAR study. There is a vaccine to MUC1 that's being presented, which has been a large effort led by Olya Finn and colleagues at the University of Pittsburgh. But I'm talking about a vaccine approach and not a CAR T approach.
Thank you. This next question is from Neil Canavan from Oncology Business Review. This is for both of you. Could you discuss the impact of COVID-19 on the conduct of clinical trials?
Yeah, so Neil, this is something that everyone is struggling with because some centers had to close clinical research altogether. I know many of my colleagues in Europe, their institutions said that there would not be clinical trial participation during the peak of the pandemic. In the US, many centers have been able to continue open, but there's been a lot of limitations by the social distancing and stay-at-home measures, where there's a lot less staff to be able to do everything that's needed to participate in clinical trials. Having said this, cancer does not go away during the COVID pandemic, and patients who didn't have other treatments and were considering clinical trials should have access to them. Many centers have made the extra effort of having less people on site doing a lot more work, so patients have options in clinical trials, and many centers have continued to be able to enroll.
Thank you, Dr. Ribas. And another question related to COVID-19 from Peter Drovert with Biotech Strategy blog. In the COVID-19 plenary session, what will we learn about the immune response to COVID-19 that may have an impact on cancer research, particularly in the field of lung cancer? Dr. Ribas, is there anything we can share at this time?
Yes, so there's going to be a presentation from a group led at the University of Milan in northern Italy, where they created a consortium in Europe that in a very short period of time captured information from 200 patients with thoracic malignancies who had COVID-19 infections. The data is embargoed until the Tuesday when this session is going to happen, but there you'll have the first ever information on exactly what you're asking. What happens to patients with pre-existing lung conditions, including cancer? Many patients with lung cancers have pre-existing comorbidities in the lung, all of which are bad prognostic factors for the course of COVID-19, and in particular, the development of acute respiratory distress syndrome. So you'll see for the first time the actual numbers of these from a pioneer group that has captured the information.
Thank you, Dr. Rivas.
I think there's a question about disparities, if they're being discussed on AACR 1 or 2 from John Ochomoke. And I want to say that disparities will be discussed in both virtual AACR meetings. It's one of the areas that the AACR has focused for many years and fostered research. For the first AACR meeting, we'll have a talk about the most pressing issue about disparities, which is what's happening in patients with different ethnic and racial backgrounds and socioeconomic backgrounds at the time of the COVID pandemic, and who have cancers and do they have different outcomes. That's going to be presented by Dr. Boyd from Memorial Sloan Kettering Cancer Center. We wanted to be sure that we had a presentation from somebody who works in a highly diverse population that would be able to capture these cutting edge information and provide it to the field. There's going to be dedicated disparities symposia at the virtual meeting too that have talks about disparities in cancer, not only disparities in COVID-19 and cancer.
Thank you, Dr. Rivas. We have a question for both of you from Mandy Jackson at Scrip. What do the toxicities look like from the combos of immunotherapies and targeted therapies, and are they manageable given the efficacy? Also, are there concerns about the cost of long-term treatment with combos of drugs that are quite expensive on their own?
Yeah, you're touching on the key questions of these. So that's why randomized trials are being performed between targeted therapies and targeted therapies adding an immunotherapy and immune checkpoint blockade inhibitor in particular, and see what is the clinical benefit. Does the addition of an immune response lead to having longer term clinical responses? So with targeted therapies, they're very active many times initially, but then the cancer can get used to the therapy. If you enlist an immune response, the immune response has memory, it remembers long term, and maybe that allows turning transient into long-term responses. Even if those results are positive randomized trials, as we'll see in breast cancer and in melanoma, but not in prostate cancer in the plenary session, even if the results are positive, then we have to see what are the drawbacks. And the drawbacks can be toxicities from keeping the therapies together, as opposed to could you get a similar benefit by sequencing or alternating the therapies. And then the economic toxicity of giving these really expensive drugs all up front. The question about patient toxicities will be addressed in the presentations. The question about economic toxicities will require other analysis of this data that I'm sure is going to be forthcoming in future meetings, maybe AACR meetings and in publications, where the benefit, the increased cost of the drugs versus the having more patients relapsing and then having complications that need to be treated and get another treatment, if one is cost benefit over the other. That we do not have results at this time.
Thank you, Dr. Rivas. This next question is from Jacob Pleath at Vantage Analysis. For either Dr. Martis or Dr. Rivas, do you have more detailed info about the allo anti-CD7 CAR T project from Gracell being presented by Dr. Wang? Do you know whether TCRA and CD7 are CRISPR knocked out in a single step? And what are your general thoughts about CD7 as a target?
So this is one of the oral presentations at the adoptive cell transfer and CAR-T session. It is designed as a universal or allogeneic CAR-T product. We have the information that's provided in the written abstract that's going to be elected in embargo. So they disrupt TCR alpha and CD7 by CRISPR. And then the idea is that if you take out the T cell receptor from the T cells, then they will not be recognizing what the T cell was naturally designed to recognize, whatever antigen that was, so that would decrease the toxicity. Then CD7 is one of the cofactors that would allow to not being recognized. So those are two steps to trying to make a T cell that doesn't attack the host and that the host does not attack the T cells so they can stand longer. This is a second generation product. They tested it. The data is early. There's 5 patients treated, 4 of them with responses in patients with leukemia, becoming negative with MRD with no residual disease. It's very early. We need to know more about the safety of these and the long-term effects.
Thank you, Dr. Ribas. We have a question, another question about COVID-19. This one from Haifei Wang with Oncology News China. This one's for Dr. Ribas. Are there studies showing that cancer patients infected with COVID-19 are more likely to become severe patients?
This is the session will present a lot of new data. We had initially Dr. Li Zhang from Wuhan presenting with a presentation. She's the only one who has published on this field, and she'll update on that results, and also on patients who have received immune checkpoint blockade therapies and have cancer. But then we just added a late-breaking series also from other colleagues at Wuhan, which is a larger series that is going to be co-published, the article in Cancer Discovery. So it's going to be the presentation and the publication of the full article, where we'll be able to see all of the data, including the tables that go with it. The data from Wuhan suggests that patients with cancer may have worse outcomes, both those two presentations, but maybe immune checkpoint blockade may not induce this worse outcome. The data is, some of it is peer-reviewed, some is not, so we have to take it as it is for now. We have 3 presentations from Europe that also capture patients with COVID infections and cancer from Italy, France, and Spain. And those 3 presentations suggest that just the diagnosis of cancer is not a bad prognostic factor. It's other comorbidities like pre-existing lung disease, the inflammatory presentation with C-reactive protein high, meaning that there's more inflammation at presentation that leads to worse outcomes. And then one of them says that recent chemotherapy also is a bad prognostic factor for the outcomes with COVID-19. Again, these are the first series. There's slightly contradicting results, and they're all have their benefits and drawbacks, and there's no comparison series in some of them. Some of them have match controls that do not have cancer, some do. So you'll have to see all of the data at the session to be able to come up with an answer to your question. And I do not have the answer to your question.
And this is Julia from the AACR. I just wanted to chime in and let you know that we will be sending out the abstracts from the COVID-19 and cancer session to registered reporters under embargo on Monday. So you'll have the opportunity to take a look at that data before it's presented on Tuesday. But the embargo is going to lift at the beginning of the session on Tuesday, which is 9 a.m. Eastern. Okay, let's see. I'm just taking a look at the questions we've got in the queue here. Another question from Lynn Peterson with Trends in Medicine. Is there a danger with liquid biopsies that they will pick up cancers too early, ones that may never become a problem, like very early prostate cancer or breast cancer that you would not treat at that stage?
So I can address that. Thanks for the question. It's a really good one. I think in some ways, and I don't mean to be glib, this would be a good problem to have, because really one of the biggest challenges right now with the liquid biopsy early detection is that we know from a variety of studies, the hardest cancers to pick up actually are ones that are at very, very early stages. So I don't think that's going to be immediately a problem per se. If you look, if you listen into the presentations in the early detection session that Tony and I were referring to early on, you will hear from both speakers that they are using each one of them different but innovative ways to help understand the location of the cancer. And so this, in some ways, minimizes or streamlines the follow-on testing that occurs clinically. The second aspect that I think is important to pay attention to in this regard in terms of early detection is really one of that both companies have looked at carefully, or both presenters rather have looked at carefully, and that is, what are the most detectable cancers? And in particular, these early efforts are tending to focus mostly on cancer types that are detected late, where the need is most pressing for early detection. So while we could worry about, too early in a context of prostate or breast, as you've wisely pointed out, I think this is probably not an immediate problem that we're going to be having.
Thank you, Dr. Martis.
Mm-hmm.
One question from Suzanne Rose at Cancer Discovery. What drug categories of cancer therapies do you think will be most promising to treat COVID-19? Why does that drug or drugs seem promising, and what can we learn about these drugs from the treatment of COVID-19 that might apply to the treatment of cancer?
So there's different categories of COVID-19 drugs that are on clinical trials. There's the ones that are trying to target the virus directly by understanding the proteins and developing drugs. That would be like the Gilead drug. That is analogous to targeted therapies for cancer, where you know that a certain protein has a certain function and develop an inhibitor for that. That would be like targeted therapy for COVID. There's drugs that have been developed to prevent COVID infections. Those would be vaccines for COVID, like vaccines for other infectious diseases like influenza, that are being then the same concepts being used for cancer. And then there's the large array of drugs that are being used to treat the consequences of COVID-19 in patients who have the bigger complications, mostly complications of respiratory distress and of hypercoagulable states. And this last category is benefiting from the experience from cancer treatment and hematological treatment for similar conditions in other settings. For example, tocilizumab, an IL-6 antibody that had been used to treat cytokine release, or CRS, from CAR-19 therapies, is now successfully being used in COVID-19 acute respiratory distress syndrome, and will have a series from Italy that accrued 300 patients in 24 hours with patients with COVID-19 infections, regardless if they had cancer or no. There's another targeted drug that was developed for cancer, JAK inhibitors. There's certain cancers that are driven by a mutation that activates JAK. But these JAK Janus kinases are downstream of cytokine receptors. If you block those, then cytokine production decreases. That's being tested in patients with COVID-19 and respiratory distress syndrome. IL-1 receptor inhibitors and several others. So this is a confluence between the field of cancer research and treatment and the field of virology and COVID-19, taking the same concepts together to the treatment of patients in an amazingly rapid time. And I hope the COVID-19 and cancer session will capture this.
Thank you very much, Dr. Ribas. That will be our final question for today. I want to thank Dr. Martis and Dr. Ribas for their insights and everyone viewing the webcast for joining us today. Again, the embargo for content in today's press conference lifts at 12:01 a.m. Eastern Time on April 27th. As I mentioned previously, there is one exception to this. The embargo for data to be presented in the COVID-19 and cancer session will lift at the beginning of that session, which is 9 a.m. Eastern on April 28th. You can access session names and abstract titles now in the online meeting planner. The link is on this slide. Full abstracts will be available at the same time that the embargoes lift. You should have received an email with embargoed abstracts for the clinical plenaries, including all the abstracts that were highlighted in this press preview. If you didn't receive that email, please contact a member of the public relations team. If you have a question that went unanswered, please reach out to a member of the public relations team and we'll facilitate offline. We can also connect you with presenters at the meeting. So this concludes the press preview for the AACR Virtual Annual Meeting 1. Thank you again for joining us and stay safe.